Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window are only visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Ashleigh Wake from Intertek.
Hello everybody, and thank you for registering today or listening today. As mentioned, my name is Ashleigh Wake and I'm Director of Business Development for Intertek Pharmaceutical Services in the UK. Prior to moving into business development, however, I have spent over 25 years working in the area of biologics characterization and quality control. In the next few minutes, I'm going to give you an introduction to some of the quality control analytics required to support inhaled and nasally delivered biotherapeutics programs. So why is inhaled and nasal delivery such an exciting prospect for biologics? There are several key advantages to these delivery routes, including target delivery to the lung, as well as providing an efficacious dose route for systemic delivery, a point which can be a particular challenge from some advanced therapy types. Although today's presentation is mainly concentrating on solutions specific to protein-based biologics, such as hormones and monoclonal antibodies, many of the fundamentals that will be discussed are also equally applicable to other advanced modality types, such as peptides, vaccines, cell and gel therapy products, as well as DNA or mRNA-based medicines. So comparing the advantages and disadvantages of nasal or inhaled delivery. Firstly, large molecules can be innately difficult to deliver systemically, as mentioned previously, at an efficacious dose level. An intranasal route avoids some of the enzymatic degradation and first pass hepatic elimination, which really reduces this barrier and can generally are what limits the biologics absorption and therefore can increase this property, contributing to a fast onset of action. More so when considering some specific indications such as treatment of Parkinson and Alzheimer's. Several biologics saw promise in this area as therapeutics, however require delivery into the central nervous system for action. Nearly all biologics substances are severely restricted from crossing the blood-brain barrier under normal conditions, and therefore delivery of these species via the nose to brain route therefore presents a real viable solution, bypassing the problems of blood-brain barrier cross. Improved patient experience and the ease of administration are both additionally significant benefits, given the tendency for the enzymatic degradation that we mentioned in the GI tract. The current typical route of administration of biologics is via subcutaneous injection or IV infusion. And these are not only less comfortable routes of administration for the patient, but it also requires the therapies to often be administered in a healthcare environment. On the downside, working with a biologic in general presents many challenges, and not excluding those of the analytics we're going to talk about. And these are things such as quite often more material being required, and this material is often incredibly expensive. There's often a greater need for troubleshooting in the development and to actually gain stability of the material. And this might necessitate the need for atypical additives to be used, for which in the worst case scenario there's no tox profile by a nasal or inhaled route. And many of these factors combined can often mean that the additional development and drug requirements simply make this type of administration financially unattractive. Once the decision has been made to develop a nasal or inhaled biologics, there are effectively two product profiles or two areas that we need to really consider through the product development in an implementing the QC strategy and the methodologies in specific needed to control. First is consider the product as any other inhaled or nasally delivered material. And this not only means in choosing the most suitable device and designing the right formulation, but importantly in the establishment of a core set of analytics to actually enable monitoring of the product, a lot of which are shown on screen. And although these are not often simple to establish, the requirements are well understood and the choice of parameters to measure are defined in regulatory guidances such as USP 601 and 1601 relevant to nebulizers, which really look to cover the release stability and product characterization. Where perhaps the new big challenge comes when considering biologics in this space is the actual introduction of analytical methods to assess control pertinent to the biologic itself. When considering product development, therefore the major challenges is to develop a product and a manufacturing process which maintains a protein 3D structure, which inhibits any aggregation, and doing all this while not compromising the product potency and safety. And really this is not an easy task. Protein structural integrity is based on relatively weak interactions, which unfortunately makes the material susceptible to change in structure. So degradation and aggregation can be caused by many features such as changes in temperature, agitation of the material, interaction of the container or the system, to name but a few. And all of these have to be thought about when actually considering the design of the device, when considering the components of the formulation, as well as the actual testing regime. For example, if we apply all this knowledge we've just learned about the tendency of a protein to degrade and what may actually induce degradation or aggregation, and we overlay this with a typical lyophilized drug to nebulizer to PMDI or dry powder inhaler type production, we can see a number of areas where potential degradation or aggregation can occur. So if we move on to specifically consider those analytics that need to be in place above those that would typically be enhanced for a nasal or inhaled product, the majority of requirements fall into three areas. Methods are required to assess aggregation, degradation, and potency. So degradants typically would include in this sense assessment of charge variants such as oxidation products, and the methods to actually assess these species, although complex, are well documented and include things such as a capillary electrophoresis, an ion exchange chromatography, or Hillick chromatography. Aggregation and potency, however, are something which require a bit greater level of understanding and something I'll come on to talk a little bit more. The final thing I'd like to specifically talk about is the assay. So although an assay to measure, for example, delivered dose is something that will be common to all products, there are certain challenges for the quantification of a biologic that really warrants further discussion, and I'll come about to talk about that in the rest of the presentation. But one major observation is due to the inherent challenges of analysing biologics in terms of the accuracy, precision, and robustness of the methodologies. It's really typical and even expected from regulatory perspective that orthogonal approaches are used to monitor the same critical quality attributes. So the techniques that I've detailed on this slide are those typical methods that are used usually in combination to assess the properties that we're talking about. The larger the bubble, if you like, the most widely applied in each area. If we move on to consider the first of these, an aggregation, it's a real expectation that this will be assessed using a stationary and a dynamic approach. For example, this may include a combination of both size exclusion chromatography and dynamic light scattering methodologies. There are, however, many disadvantages and advantages of each of the approaches that you can use, all of which I've kind of highlighted on screen for the main techniques. And as an example, SEC has significant advantage in its use and it produces a quantitative result of what is acceptable accuracy and precision, using techniques and equipment that is methodology that is common to most laboratories. However, the interaction of the product with the column, the solvent, it can actually lead to low recoveries of material or, importantly, the formation of aggregates giving false results. So during method development and validation, it's therefore really typical if SEC is to be used as the final release or quality control method, Other analysis has been performed by something like AUC, which is an in situ technique to actually check out the process of separation isn't inducing any aggregation or low recovery. As I mentioned, quantification of a protein as a delivered dose is something that on phased observation may appear incredibly simple. For assays of proteins, it's not typical that chromatographic approaches akin to small molecules are used, but rather concentration is determined using a non-specific total protein assay in its simplest form, something as absorbance at 280 nanometers. Assuming, however, that a suitable quantitative method is established, one of the greater challenges can actually be determining the delivered dose might be to do to inherent tendency of proteins to absorb onto surfaces, for example, the surfaces of the device, for the surfaces of the sampling equipment. The extent of adherence that you actually get is specific to an individual protein, it's specific to the protein concentration and the surface on which you're actually dealing with. And any slight changes to one or more of those can actually lead to differences in the extent of adhesion. There are several ways to avoid this. For example, you can use specialized products in process where possible, or you can pre-coat any surfaces with a surfactant or a blocking protein. So this is something like BSA, which will effectively flood the surface. Similarly, these materials could be actually added to the extraction buffer. However, depending on the approach that you have to use to recover the material and the actual components that you are including in that process may actually drive the assay method that you're going to have to use. For example, if you use BSA to actually coat or to extract protein in the delivered dose method, the A280 or the colorimetric approaches won't work, and you'd need to have an ELISA or an LCMS approach established. Potency assays are complex and probably the most complex across all different types of quality control methods, regardless of modality or purpose that you can actually put in place. What I've tried to do here is present the very simplest definition of what an actual potency assay is, how it works, what it does. But I really apologise to any cell biologists in the audience, as this is perhaps the most oversimplified thing in the whole entire world. So in the very most basis, a cell line is chosen based on an alignment or based on the mode of action of the actual product, which is cultured in a laboratory setting. The target test material is then applied over an 8 to 12 point dilution series, which actually is there to affect one of the more biochemical processes in the cells, which will either reduce or increase the expression and therefore the concentration of a target or marker protein. This target or marker protein can be quantified by ELISA, and from this and comparison to a reference material, the potency or the relative potency established. So calculation of potency is really based on comparison of the response of the test material to that of a well-defined reference material. So this reference material is something which has been well characterized and typically would have been used in a clinical setting. The response of the two are plotted, the parallelism is compared, and then the relative potency based on something like comparison of the EC50 of both materials. This is often a challenge, and as I said, accepting criteria and accepting limits are often very different to what would have been seen in typical chemical assays. However, understanding that you're keeping your potency through nebulization, through nasal formulation, is really probably one of the most important features that you could possibly address in these type of products. So in summary, there are real advantages to nasal or inhaled delivery for biologics, specifically in this instance proteins. And advantages that could overcome some of the challenges that are seen with achieving systemic bioavailability or in getting the product to the target areas such as the central nervous system. Product development strategies therefore require establishment of not only those methodologies that are traditionally associated with nasal or inhaled products to support development and ensure quality through development, release, stability, etc. But also you need to include those methods that actually address the quality control attributes of the biologic itself, so specifically aggregation and potency, as well as some of the degradation methods, such as the ones we talked about for oxidation products. These methods are inherently less complex and less accurate and precise than their small molecule counterparts. And often there's a requirement to use orthogonal approaches to ensure the very best quality and consistency of product. So thank you very much. Thank you for listening, and I'd be delighted to take any questions.
Okay, great. So the first question we have for Ashleigh is, what are the major challenges to a successful development of an inhaled biologic?
Yeah, obviously there's quite a few challenges that you could talk about there, but some of the major things that we need to look at is making sure the formulation supports actually maintaining the integrity of the biologic through the inhalation process. It's making sure as well that any excipients that we need to put in the formulation are kind of compatible with the kind of inhalation route. If they're not, it's not a no-no. It just kind of means that we have to think about doing some toxicology trials to actually show that the placebo or the excipients aren't harmful. So that's a major challenge, to make sure you kind of get that part right in the first place. The next challenges can really come with, getting the material out of the device. So as we know, proteins are incredibly sticky. A lot of the surfaces that they come into contact with in the devices can actually cause the proteins to bind. So again, this comes back to excipient choice, but also the actual device selection itself can aid in there. So I would say that they're probably the two major challenges that we face.
Okay, and how is reproducibility of doses ascertained?
Yeah, so again, it's a good question, and it's something you have to kind of work at sometimes to make sure your formulation, your device, etc., actually gives you a reproducible dose. So in terms of the measurement, it's actually quite simple. So we collect essentially the inhalation, the mist, the spray, whatever we're looking at. If it's a nasal device, it would be just the same through an actuation. And we actually collect all the material that's gathered and actually just then perform a simple assay on the material that we've got. The only kind of downside to that, I guess, is that the volumes that you're collecting in can sometimes be quite large. So if you have a small dosage amount, detection limits can be a bit of a challenge, and you might have to move away from kind of the traditional UV-based assay methods you would tend to use in the QC program to something more sophisticated. But it is just a process of actually, firing the device, collecting the inhalation, collecting the mist, collecting the spray over the time that you would give the dosage to the patient in a minimum of, say, 10 replicates from 10 different firings, and then looking at the variability across that.
And have there been any recent developments for the assay for recovery?
Yeah, so the assay itself is we tend to, or try to keep it very simple. So doing something like an A280 even at the end. The actual recovery, I guess, is the challenge in terms of recovering the material from the devices themselves to make sure we're actually accounting for like a full mass balance. So there are a kind of few approaches that we've developed, which is to do with the actual diluent or kind of recovery solution. So adding additives in there. There are approaches that you can do if you're drastic by kind of pre-coating the surfaces, etc., or adding something like BSA into the formulation itself, which brings on other challenges. But a lot of it is, to do with actually recovering from the devices, and a combination of solvents, salts, pH, inorganic ions and cations, things like that you can add that are kind of specific to what you're looking at in order to aid recovery. And typically, it takes a little bit of work, a little bit of trial and effort, but typically good recoveries, kind of the 90% pluses can be achieved.
Okay, it looks like we have time for just one more question. If we didn't get to your question, we'll be passing those on to Ashleigh, and she can follow with you directly. So please continue to type in your questions. But the last question for the webcast is, how do you select techniques for aggregation?
Yeah, so again, it's quite a complicated question. So in development, certainly, until you actually know your protein, I'd be applying a number of techniques, so an orthogonal approach. So typically something like SEC with or without mild detection and or AUC are the two you would go for. In that sense, the AUC is really kind of validating what you're seeing from the SEC. So it's making sure that on a methodology basis, you're very confident that any aggregation you're seeing by SEC is inherent in the product itself and not just an excipient. As you move further through the quality control program, it may be that you get enough confidence built up to maybe drop one of your orthogonal approaches. And then if you had to pick just one standalone that was kind of, readily available in all QC labs, I would go for SEC. But I guess to me, the ultimate for this type of work is probably something like AUC. But we try to make sure that whatever methodology you choose or put in place is actually going to give you kind of the longevity of your pipeline.
Okay, well, thank you, Ashleigh.
Thank you.
And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website. And as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.
Bye.